More links, the same exact links from before. I recommend watching them. So why do we care about sequence alignment? Why do we care about identity? Why do we care about similarity? What can we do with this stuff? So we can use sequence alignment algorithms. The last video on that link actually talks a little bit about using BLAST or L-Align, which are some alignment algorithm softwares freely available on the internet. You punch in your sequences and it will align them and spit out identity and similarity for you. You guys don't have to do it, but I've made a comment for winter that we'll definitely be playing around with BLAST and L-Align. But I invite you to go look at them if you want. But the one video, the guy walks through it and shows the output from these, and it's pretty cool. So it would let you look for related sequences, but more importantly, like it would let you look at common structures. It would help you identify those domains, those motifs, those unique repeating features. The other thing you could look up is you can do this with DNA sequences and nucleotides as opposed to the amino acid sequence. And so this kind of concept of sequence alignment, identity, and similarity forms the basis of finding your relatives, genetic relationships, and how similar your genes are. You can use similar algorithms are used. Okay. And so when you have sort of a % of identical residues, and you have, as you increase the length of the alignment that matches, you get into this region where you know you're going to have really structurally similar proteins. And so when you have a protein that you don't really know what it does and you don't really know its structure, but you can compare it to something that is already in a bank of known proteins, and you spit out that it is very similar, it has really high % identity, really high % similarity, then you can start to speed up your prediction of what its structure looks like and possibly what it does. And we're definitely still isolating proteins from the body, especially when we're talking about trying to figure out what's wrong with a protein that's misfolded and trying to figure out where the sequence is swapped and what is happening when there's a problem. But also from other species, we can go ahead and we're still characterizing proteins all the time. Like, they're not done. We don't know them all. But we do have some good banks that have a lot of common structures and common proteins available for you. Protein Data Bank or PDB, CATH or Gene3D.